Welcome viewers to this segment of online lectures. Here we are going to discuss Charophyta. Charophyta is a division of freshwater algae. It's a group of complex green algae that form a sister group to Chlorophyta. It's a group of eukaryotic, photoautotrophic, aquatic organisms having cellular level of organization. It is included in domain Eukarya and kingdom Plantae, with the cladistic inclusion of the Embryophyta. The Charophyta is synonymous to Streptophyta. So far as distinctive features of the group are concerned, the algae belonging to this group are multicellular and complex. Their main plant body is haploid and known as gametophyte. It lacks differentiation into root, stem and leaves and is called thallus. Like Chlorophyta, the thallus looks green due to dominance of chlorophyll pigments. Cells possess chloroplasts having chlorophyll A and B as the main photosynthetic pigments. Phycobilins seen in other groups of algae are absent. Unlike chlorophytes, their cells have enzymes similar to higher plants. They possess aldolases, copper and zinc superoxide dimutase, glycolate oxidase, and flagellar peroxidase, which makes them similar to embryophytes. Next important feature of these algae is that they store food as starch, which is localized in plastids as higher plants do. The Charophyta and Embryophyta together form the Streptophyta, excluding Chlorophyta. Unlike Chlorophyta, within chloroplasts you don't see pyrenoids, which makes them closer to higher plants. Flagellar immotile cells, if present, are lateral in position. Cells use phragmoplasts during mitosis. So far as cell structure is concerned, cells have a well-defined cell wall, followed by plasma membrane. Inside plasma membrane there is colorless fluid called protoplasm. Protoplasm is well compartmentalized by endomembrane system. Therefore, the cells of charophytes have non-membranous as well as membrane-bound organelles. The non-membranous organelles are ribosomes, centrioles, and basal bodies. Single membrane-bound cell organelles include endoplasmic reticulum, dictyosomes, lysosomes, and microbodies. Double membrane bound organelles include mitochondria, chloroplast, and a well-defined nucleus having nuclear membrane and chromatin material. Cytoplasm possesses ribosomes of ATS type, typical eukaryotic character. There are numerous small discoid chloroplasts which are disposed around the periphery of the cells. No pyrenoids are seen. The internodal cells are sometimes multinucleated and their nuclei often possess large nucleoli and scanty chromatin. In these cells, cytoplasm forms only peripheral layer with a large central vacuole. The cell walls are composed of cellulose, though there may be also superficial layers of more gelatinous material of unknown composition, mostly glycans, glucans, mannans, etc. The stored food inside cells is always starch, except in oospore, where oil is seen. The starch also accumulates in special storage structures termed as bulbils, which consist of rounded cells. The cells have varying size and are developed in clusters on the layer of main body axis and rhizoidal regions. They are mainly developed when algae are growing in fine, slimy mud. Cytoplasmic streaming is seen around the cells and is well visualized in the cells of internodal regions in the axis. Reproduction, it occurs through vegetative means as well as sexual means. The reproductive organs in this group of algae show high degree of specialization. They are multicellular, jacketed. Female sex organ is large oval structure with an envelope of spirally arranged green filaments of cells. It is termed as oogonium. The male organ is also large, bright yellow or red in color, spherical in shape, and is usually called anthedium, although some workers regard it as multiple structure rather than single organ. The sex organs are developed in pairs from adaxial side of nodal cell at upper nodes of primary lateral branch. Oogonium is formed above anthedium. They are sufficiently large to be easily seen by naked eye. The algae are also visible to naked eye, especially the bright orange or red anthedium. It is visible to naked eye. Many species are dioecious, having separate male and female gametophyte. In others, they are monoecious, that is, born in the same individual. Thus, anthedium and the oogonium, they develop at different times, preventing fertilization. Anthedium produces flagellated male gametes called anthrozoids, whereas oogonia produces egg cells. In case of two types of sex organs arise from different points of lateral branch, the time duration is different. Fertilization is always external and supported by water, as male and female gametes are released into water. The fusion of male and female gametes is oogamous or sometimes anisogamous. Zygote formed during fertilization undergoes zygotic meiosis followed by mitosis that leads to the development of gametophyte. So far as vegetative reproduction is concerned, it occurs readily in corals by different methods. One method is formation of protonema. Protonema are filamentous structures thread-like structures formed by the thallus of charates. They can be primary or secondary. Primary protonema is formed directly from the thallus, whereas secondary protonema is produced from primary protonema as a result of mitotic division of cells. Secondary protonema may develop even more rapidly than primary ones. The fragments of nodal regions of the thallus have dominant cells, and these cells, after hibernation of the plant, divide mitotically and produce the whole thallus along with the root-like structures called rhizoids. It is probably this power of vegetative reproduction which explains the fact that the species of Caraceae are generally found forming dense clonal mats in the beds of ponds or lakes. Sometimes you will see these in streams covering large areas. So far as classification is concerned, Charophyta includes Charophyceae, which includes 6 genera. The most prominent genera in the group is Chara. The others include Nightella. Tulipella is another genus which is very commonly seen in freshwater bodies. We will now try to understand about genus Chara. Chara is a green alga belonging to division Charophyta. It is seen in freshwater bodies, and its systematic position is domain Eukaryota, then kingdom Plantae, division Phycophyta or algae, group Charophyta. However, modern biologists like Lillard et al. In 2012, they have divided Chara into a separate group, separate division called Charophyta. Chara is a genus of charophyte, green algae, in the family Caraceae. The genera included in it are multicellular and superficially resemble land plants because of stem-like and leaf-like structures. They are found in freshwater, particularly in limestone areas, throughout the northern temperate zone, where they grow submerged, attached to muddy bottom. They prefer less oxygenated and hard water and are not found in waters where mosquito larvae are present. They are covered with calcium carbonate deposits and are commonly known as stoneworts because of their hard structure. Cyanobacteria have been found growing as epiphytes on the surfaces of Chara, where they may be involved in fixing nitrogen, which is important to plant nutrition. So far as structure is concerned, the gametophyte of Chara has a branching system which is complex with branches derived from apical cells which cut off segments at base to form nodal and internodal cells alternately. The main axis bear whorls of branches in a superficial resemblance to equisetum. Equisetum is a pteridophyte. They are typically anchored to the littoral substrate by means of branching underground rhizoids. Chara plants are rough to the touch because of deposited calcium salts on their cell wall. The metabolic processes associated with this deposition often give Chara plants a distinctive and unpleasant smell of hydrogen sulfide. So far as morphological features are concerned, the plant body is gametophyte and consists of main axis differentiated into nodes and internodes. It has dimorphic branches, that is long branches of unlimited growth and short branches of limited growth. The main axis has root-like structures called rhizoids, which are multicellular with oblique septae. There are stipulodes, that is needle-shaped structures at the base of secondary laterals. After having studied its structural details, let us now study reproduction in Chara. Chara shows both vegetative as well as sexual reproduction. Vegetative reproduction, it generally occurs during favorable conditions and takes place by following methods. Number 1, bulbils. Number 2, tubers. Number 3, formation of secondary protonema. 4th, amylum stars. First, bulbils. They are multicellular, rounded or ovoid reproductive bodies formed at nodes of primary laterals or rhizoids. Chara baltica produces bulbils at nodes of primary laterals, whereas Chara aspera produces bulbils on rhizoids. After detachment from parent plant, they grow into new thalli, tubers or amorphous bulbils. They are multicellular, lateral outgrowths formed on the nodes of main axis or rhizoids. They possess loose aggregation of cells and are seen in Chara baltica, Chara frigifera and Chara delsecatula. Now, secondary protonema. They are tubular multicellular bodies produced from rhizoidal nodes or from basal cell of primary rhizoid during favorable conditions. After detachment, they grow into new young plants. Now, amylum stars. They are clusters of cells with star-like appearance, born at basal nodes of main axis. On detachment from parent plant, they grow into new young plants. Now, sexual reproduction. Chara reproduces sexually, generally in response to unfavorable conditions. It occurs with the help of definite multicellular reproductive bodies called globules and nucleus. Most species are monoecious and homothallic, but few species are dioecious and heterothallic. For example, Chara wallichii. First, globules. They are spherical, multicellular, male reproductive bodies born at the nodal regions of primary laterals. Each mature globule is bright yellow or red in color, having enthedial filaments surrounded by shield cells. The cells in enthedial filaments form male gametes called entherozoids. Each entherozoid is a helical, biflagellated cell that, after being released, actively swims in water towards egg cells. Now, nucleus. They are multicellular, oblong, female reproductive bodies born at nodal regions of primary laterals above globules. Each nucleus has a pedicel cell for attachment with the nodal cell, a stalk cell, a large egg cell that remains surrounded by 5 long spirally twisted tube cells and coronary cells situated on the top of tube cells. Now fertilization. Cara shows oogamous fertilization dependent on water. It occurs inside nucleus and leads to the formation of a diploid cell called oospore. At the time of fertilization, the tube cells in nucleus near coronary cells separate slightly from one another, which leads to the formation of 5 narrow slits or apertures. These slits remain filled with a colorless viscous fluid. Enterozoids released by globules swim through water towards nucleus and stick to the fluid. One enterozoid penetrates the ovum near receptive spot and fertilizes it to form oospore. Oospore is a hard, nut-like, yellow, red or black diploid cell with oval or ellipsoid shape. It has a thick 4-layered cell wall. After passing through unfavorable conditions, it becomes active with the onset of favorable conditions. Now, germination of oospore. As favorable conditions approach, oospore becomes active. The diploid nucleus present in its cytoplasm moves upwards and undergoes meiosis to form 4 haploid nuclei. 3 nuclei degenerate and the 4th one remains functional. It undergoes meiosis to form a rhizoidal initial and a protonemal initial. Rhizoidal initial divides further by mitosis to form a rhizoid, whereas protonemal cell gives rise to an upright filament called primary protonema. Primary protonema differentiates into nodes and internodes and forms laterals to form a haploid plant body. Now, so far as life cycle of Cara is concerned, Cara shows haplontic life cycle characterized by formation of mitogametes followed by syngamy and zygotic meiosis. Syngamy is oogamous. Only zygote या oospore represents the diploid stage in the life cycle. This is all about structural details and reproduction of Cara. Hope you have understood the lecture. Thank you.